Só um minutinho que ele vai dar um aviso.
Tá, acho
É,
que chegou
pronto.
aqui. Ótimo. So thank you everybody for joining our immuno webinars of October 7th. So we had the pleasure of receiving Dr. Cecília Pessoa Rodrigues. Dr. Cecília is now working at Roche, Basel, Switzerland, as a postdoctoral researcher at the Department of Immunology. So she just transitioned from academia to industry. So it's a pleasure for us having you here, Cecilia. So the stage is yours. She's going to present. She sent me a title of her talk as Macrophage Quality Control Regulates Hematopoietic Stem Cell Clonality. And now she changed a little bit her title here, but she's going to talk about this great subject for us. Thank you very much, Cecilia.
Yes, no, or the topic's the same. I'm actually extremely happy to be here today. Diego didn't mention, but I'm actually an alumni from the program in USP. So for me, it's really an honor to see some friendly faces today and talking about some things I've done in my postdoc. So as Diego mentioned, I am located now in Basel, but today I'll be talking about my first postdoc that was conducted in the Zone Lab at Harvard and also in Boston Children's Hospital, where we are looking at macrophages and how macrophages can mediate and regulate stem cell clonality. And you might wonder, why on earth me as immunologist, I care about hematopoiesis? What is that about? So in a nutshell, hematopoiesis is basically the origin, so where our favorite cells are coming from. So our favorite cells from the immune cell system, like neutrophils, macrophages, DCs, NK, you name it, they all stem from one cell, that is the HSCs, aka hematopoietic stem cell. For many, many years, we thought that this was a stepwise model where one stem cell would then differentiate in a very much hierarchical model into more differentiated cell types. But nowadays, we know that this stepwise model, it's actually not really what's happening in reality. In fact, single-cell technology showed us that what's happening in reality is more like a continuous model, where you have HSCs, so hematopoietic stem cells, and I will call it from now on as HSCs, or HSPCs for hematopoietic stem and progenitor cells. So what you see is that they are actually a pool of cells, very heterogeneous, and then there is a continuous differentiation towards a more specialized cell. And cell here meaning every single cell type, cellular component that we have in our blood, including our favorites. And moreover, what we found is that this process is a clonal event, meaning that you have different clones with different dynamics. And for some reason, a clone continues and propagates into a very final cell type, while other clones, they stay in the marrow. And this begs the question, why? Why somehow we are selecting good clones to stay and others not? And in the Zone Lab, we're very much focused on trying to understand that, because this could be a way of alleviating a bunch of clonal diseases like leukemia, for instance, or other blood disorders. And in fact, in the lab, we use zebrafish as a model because we see a very nice correlation between the human system and zebrafish. And I don't have to tell you that they are very much easier to imagine. They are transparent as a neighbor, so you can really see what's happening with the cells. You just put them under a microscope and you can trace the cells. So a very talented PhD student in the lab, he observed that stem cells and macrophages, they interacted. And actually, they interact in a very, very special condition. So we have two flavors. Here you see the stem cell in magenta and the macrophage in green. And I hope you can appreciate that in this particular example here, macrophage will interact with stem cell and then fully engulf the stem cell. It will doomed. So that clone is diminished. It's not there anymore. While here, a macrophage will come, interact with the stem cell, take parts of the cytosol, and just after this interaction, the stem cell goes on and divide. And because of this phenotype that the macrophage comes and sample parts of the stem cell, we're calling this phenotype as grooming. Sam, the PhD student, went one step forward and he identified the molecule that was mediating this interaction of the macrophage and the stem cell. And this molecule was caroticlin, which from now onwards, I will call as CALR. So CALR is actually an ER protein, it's a chaperone But in some particular cases, it travels to the surface of the cell and it works as an eat me signal. So what Sam discovered is that CALR was important for this interaction, but not only for the eating, but also for the grooming, the don't eat me, where it samples parts. And this was very interesting because it was the same molecule. And once you deleted it, you would affect both grooming and dooming. In this very first paper, we were able to also validate that the levels of CALR could somehow be a predictor of grooming or dooming. And to be more precise, what we found is that the levels of reactive oxygen species, so ROS, was also correlating to the levels of CALR. So the more stressed the stem cell was, the likelihood that it would be eaten, it was higher. Nonetheless, this begs the question, is it really a matter of balancing in the sense that if you have a lot of CALR on the surface, you're going to be doomed, while if you have low levels of CALR, you're going to be groomed? Or it's actually a double signal, like you have CALR that mediates the interaction, and there's something else that tells the macrophage like eat me or don't eat me. And this was the basis of my project when I started in the lab, trying to understand which signals it was giving, the stem cell was giving to the macrophage to eat me or don't eat me. And to tackle this question, what I first did was to do a chemical screen where we're looking at a bunch of chemicals. So to be honest, it's 1,200 compounds. And we're looking at not only ROS levels, where we're using cell ROS to have an idea. So it's a cell probe. It's a probe that enters the cell when it's alive. So we don't have to permeabilize the cell or create more stress on it. And we're also looking at the CALR levels. And here, to be very sure that we are only looking at the CALR levels on the surface of the cell, we're not only looking at the immune staining, so we're using antibodies for that, but we're also using a strategy that's named split turbo ID. So it's a surface construct where we have the N terminal, we have CALR, and the C terminal of this molecule, we have a membrane protein. And here I am using ECAD. So the idea is that when there's proximity of protein A, CALR, with ECAD, so a membrane protein, we have the full reconstruction of the split turbo ID, and the neighboring protein will get biotinylated. And then with streptoavidin, we can see the expression only on the surface. And here, just to show you an example. So in the first row, you see a construct that has the surface CALR. So you barely see beautiful decorating the surface of the cell. While if we have only the ER CALR, you see it more punctate phenotype, and it's cytosolic. Okay, with that then, we went forward, we did the screen, and what I identified is that among these 1,200 compounds, only 93 upregulated CALR on the surface of the cells. Then I took the next step, which was, okay, so these compounds, they upregulate CALR, but what they do with the macrophage stem cell interaction, is it also affecting? And obvious answer, no, it's not all the 93 compounds that also affect the interaction between macrophages and stem cells. And again, here we're using fish as a model, and macrophages we're tagging with MPEG. So every time you see MPEG, it's a macrophage marker, and RUNX1 is a progenitor marker. And what we found is that 22 compounds, they also upregulated the interaction with macrophages and stem cell. Then the next step was trying to deconvolute if there was any difference between the ROS-independent and ROS-dependent compounds. And for our surprise, we found a very striking difference in the sense that the ROS-dependent compounds, they promote only or mainly dooming. So they would doom the cell that was stressed, as we predict. But for our surprise, we see that the ROS-independent compounds, they were promoting grooming. And why is this so surprising, you might ask? Well, because we are seeing high levels of interactions, we see high levels of surface CALR. So this first hypothesis that it was only the CALR levels that was distinguishing the dooming versus the grooming, it was wrong. Because these cells, they have high levels of surface CALR tickling, and yet we are seeing more grooming instead of dooming. So then our first hypothesis of seeing this result was, well, probably these 6 compounds that is ROS independent, it is somehow affecting the macrophage. So these macrophages, they're not proper activated somehow. and ergo they're not doing the role of engulfing the cells, and ergo we're seeing more grooming. With that, then my student Catherine, she took the effort of testing the macrophages function upon a stimulation of the 6 compounds that was ROS independent. And what she found is that our ROS independent compounds, they did not affect migration. So macrophages, they could migrate towards LPS. They were pretty nice and working macrophages. And when she looked at phagocytosis, so she is using here phagocytosis beads of E. coli. So it is E. coli rhodo that will change color in a very acid environment. She saw no difference. So this tells us that our compounds, they were not really affecting the macrophage function. Macrophages were able to do phagocytosis. It was not that, it was something else. So we were really puzzled about that, and we were trying to find what was the something else. And to address this question, what we have done was a CRISPR-Cas9 screen, where we tried to identify the genes that were regulating surface CALR. So here's a little bit complicated to see because it's the inverse. Since it's a knockout CRISPR, we're looking at after stimulation, the genes that are downregulated, because these genes will be important to upregulate CALR, because it's a knockout screen. And to have an idea that was a surface CALR in a ROS independent context, we are not only using normal cells, but also cells stimulated with one of these ROS independent compounds, which is DLPPMP. That for now onwards is our bait for testing the ROS independent compounds. And among the markers, we found a very strong type 1 interferon response that was important to induce surface calreticulin in the surface of the cells in the ROS independent context. And among the targets, we found Tol3. And me as an immunologist by training, I was very happy to see a very familiar molecule, because that's a DS RNA sensing that we know it's important for virus response, but yet we had to validate, is it real what we're seeing? And to validate that, we took advantage again of the fish, and we depleted Tol3 using morpholinos. And what we found is that depletion of Tol3 actually indeed reduced the levels of CALR in the progenitor cells, here depicted by RUNX1. And this also holds true when we use a compound. So we're using here a compound that blocks the interaction of the DS RNA to the sensor. And we again see a decrease in the surface. And more importantly, when we look at the interaction type, we see that depleting Tol3, we also promote more dooming. So in fact, suggesting that this pathway somehow is affecting the dooming versus grooming. Nonetheless, if you're very picky, if you're review number 2, you're gonna tell me, well, Cecilia, both morpholinos and your compounds, you have fish, it's a global effect. So how can you be absolutely sure that you're not affecting the whole system, and ergo you're seeing these differences in eat me and don't eat me? And to tackle this question, what we have done was parabiosis experiment. So we stitched 2 embryos of fish. One embryo had the progenitor cells labeled in RUNX1 and the macrophages labeled in PFP. And the other embryo, it was only macrophages labeled with MPEG. So we had 3 color system. And one fish we treated with our standard morpholinos, so nothing was depleted. And the second group, we had the Tol3 morpholinos, so our mutants. And what we found is that regardless of the source of the macrophages, if it was a macrophage that had Tol3 or not, they interact more with stem cells. And when we look at the type of interaction, they were more prone to doom the stem cell, regardless of the source of the macrophage. So it was not an effect on the macrophage, it was something on the stem cell that was signaling through the macrophage. And we were very interested to understand what was that. But before that, again, you can be picky and tell me, well, you're only looking at the fish. Maybe in mammals, it's something completely different. We have a complete different response, and ergo, all your results, they are nonsense. And to understand if it was conserved throughout evolution, what we have done was to turn a little bit, so stop using the fish and we turn to the mouse. And we use an in vitro system where we took bone marrow, and we had bone marrow-derived macrophages, and we co-cultured them with mouse progenitor cells. Here we're calling as LSK, so lineage SCA1 positive, KIT positive. And these progenitor cells, this LSK, we treated or not with poly-I, poly-C, which is a Tol3 agonist. So we co-culture them, and it's autologous. So it's the same mouse, same macrophage from the same mouse, same progenitor from the same mouse. We co-culture them in a 2D system, and we ask which type of interactions they were having. And I hope you can appreciate that in the control, nothing much is happening. So you have macrophages here, they interact, but like long interactions, nothing much. But when we pre-treated the progenitor cells with poly-C, you start seeing a lot of grooming events here. And when we quantify, we indeed see a significant increase of grooming. So suggesting that our phenotype was not only conserved throughout evolution, but also the mechanism was most likely conserved throughout evolution. And again, I took advantage of my background, and I was thinking, well, poly-IC is leading to this grooming. I'm seeing this increase Tol3 influence in CALR. Well, what do I know about Tol3? I know that Tol3 could be a mimicking of a virus infection or something. What do I know about virus? Well, I know that when a cell is infected by virus, they upregulated beta-2M. And I'm just taking a long story short. This was an important thought because beta-2M, so beta-2 microglobin, is an essential component of class 1 molecules. And it's known in the literature that beta-2M works as a don't eat me signal. So with that in mind, I thought, well, what if it's a double switch mechanism where you have CALR, that is the eat me signal, and promotes the interaction. But if you have beta-2M or class 1 molecule being expressed, this stops the macrophage to eat the stem cell. So working as a don't eat me signal. So you have the interaction, but you don't eat, and then you have grooming. And this was the hypothesis that I started to test it. And first, what I did was to generate crispins. So this is a mosaic deletion of beta-2M in the fish. And I look at interaction. I don't see increase or decrease in interaction. Nonetheless, when I look at the type of interaction in the beta-2 crispins fish, I do see an increasing dooming, suggesting that the model might be true. Then I took the next step, which was, okay, I have these molecules that they promote grooming in Y-type conditions. What if I take out beta-2M? What this means? This means that I would have CALR being expressed, I would have interaction, because this is what I see in the Y-type situation, but I don't have this molecule that was stopping the eat process. And in fact, it's exactly what happened. So if we take here DL-PPMP as an example, in the Y-type situation, you see that the black bar is bigger, so suggesting there's more grooming. But when we use this in the depleted beta-2M, that here is depicted by this small b letter here, we see increasing dooming, suggesting that indeed beta-2M was acting as a stop me. Again, that's a mosaic deletion. So there's still some Y-type cells therein. We had to be absolutely sure that our phenotype was real and not just a side effect of having a mosaic situation. So to this end, we generated the fish line. So it's a full knockout of beta-2M. We validate, so you see here by fax that there was a global deletion of beta-2M significant. And again, when we look at the type of interactions of stem cells with macrophages, we see that there was a decrease in the grooming phenotype while an increase in the dooming. So the stem cells, they were being more eaten in our fish. Okay, so we were convinced that beta-2M works as a don't eat signal, and cells expressing beta-2M, they're not eaten. Okay, so our next question was that if we have this increase in dooming, so if we're taking more clones out, what happens to stem cell clonality? Does it affect it? Do we see decrease in clonality complexity, or nothing changes? And to tackle this question, we use a fish that we have in the lab. The name is Zibo. So Zibo, again, long story short, is a fish that has a UB cassette that has 3 colors and dracoline Cre recombinase. And in the presence of Cas9, you start chopping this LOXP sites and you have different color combinations. So you have up to 30 color combinations. And depending on when in the time of development you hit them with the Cas9, you can really trace back the clones based on the colors, because the same clone will have the same color. So you can go back in time and say, like, this clone, this yellow clone gave rise to these myeloid cells, or this blue clone gave give rise to that lymphoid cells. So we took advantage of the system, and what we did was to use then Twister and SIBO, where we induced the depletion 24 hours after post-fertilization, which is when blood starts to be developed. And we use that in the Y-type system, where we just CRISPR out a desert region in the genome, so not affecting any response, but we have the cut. Or we deleted beta-2M. And that's cool because, again, it's a mosaic situation. So you can have the clonal competition where you have Y-type and deleted cells in the same marrow. And you see how the clones compete against each other. And that's very powerful. You're going to see why. And also we had or not depleted the macrophages. So first things first, let's see what's looked to beta-2M. So when we look at the beta-2M clone composition, you would see that there was a high Gini coefficient. So Gini coefficient shows how different clones are to each other. And the closer to one they are, the lower differences you see. So you see that the Gini coefficient here, when you deplete beta-2M, is very low. So the clonal complexity is very, very small compared to the Y-type. And on top of that, when we look at the clone numbers, it's also decreased. Nonetheless, when you see the cluster size, we see that there is a significant increase in the beta-2M. So these three graphs together tell us that the clonal complexity in the beta-2M knockout is lower, and most likely you have few clones that are taking over the marrow. So you have lower complexity, low number of cells, but a dominance. So there's one clone that is dominating everything. And importantly, when we deplete the macrophages, so you see here by fludarate treatment, you see that we rescue the Gini coefficient. So it goes back to the Y-type levels. We don't rescue the clonal numbers, but this we were somehow expecting because we don't have the interaction with the macrophage. So every type of interaction is lacking, including grooming and great stance. So we don't have the number of clones being rescued, but we do rescue clonal dominance. So it's not that we have fewer clones that take over the marrow, but it's more homogeneous. We were very interested to understand if indeed, or what was the phenotype and the genotype of this clone that was taking over the marrow. And to this end, we sorted by facts the dominant clones and the non-dominant clones, and we asked, what about their genotype? Are they Y-type cells or are they mutant cells? And what we found is that the dominant clones, they were Y-type cells, while the non-dominant clones, they were mutant cells. What tells us is that the Y-type cells, they have beta-2 in their surface, so they survive the macrophage interaction. And it's not that they are taking over the marrow, but they are surviving actually. So it's not they are massively proliferating, they are just surviving the macrophage dooming, and therefore they make the marrow. While when you don't have beta-2, you are doomed and you don't propagate your color. And this is exactly what we're seeing. So then we thought, well, that's very cool. Can we have an idea of the molecular bias or what is the molecular structure that is giving rise to this beta-2M in the surface? So we thought, well, what if we go backwards? So we know that beta-2M is important, and we know that clonality is important. So what if we do ZIBOs and CRISPRs and Twister in the RF3 mutant? RF3 is a transcription factor that it triggers beta-2M expression. And more importantly, what would happen if we do the same exercise with a TOL3, a mutant? Can we see that TOL3, RF3, and beta-2M affects stem cell clonality? And in fact, when we do these experiments, this is exactly what we see. So when we affect RF3, so when we deplete RF3, when we deplete TOL3, we see a decrease in clone numbers, we see clonal dominance, and when we sort the dominance, we see that there are Y-type cells. And more importantly, again, you can tell me, well, Cecília, that's fish. What about mouse? So I'm very pleased to say that in the mouse, not only fetal liver that was published before, but actually also in adult. So here you're seeing a bone marrow from a living mouse. And in green, we see stem cells, and in red, we see macrophages. They do interact. And more importantly, when we treat them with the ROS independent compound, so this DL-PPMP, we see increase in stem cell proliferation, which is a proxy for grooming. And we also see an increase in class 1, MHC class 1, which is one of the components that beta-2M makes a part of. So with that, I'm come to the summary of part 1, which I hope I convinced you that beta-2M counterbalance CALR, ITMI signal. Beta-2 mutants, they show increased dooming. Beta-2 crispants, they displayed lower myeloid and lymphoid clone complexity. And the beta-2 dominant clones, or RF3, Pol3 dominant clones, they're actually Y-type cells. And here I'm making a pause because also in the project we had a pause. And it's that moment as a scientist where you feel a little bit like a detective. You're like Tintin, where you try to understand, well, I'm having a virus response, like a classic virus response here, in a system that I'm pretty sure is not they're not infected by virus. I'm pretty sure the fish are fine. They go to the vet, we test the water. They're not infected. So what can be the endogenous trigger that is leading to this beta-2M that is also conserved throughout evolution, because we're seeing the mouse. So it's something that has to be around for a while and endogenous. And with that in mind, we first wanted to double check if it was indeed a virus response or like mimicking a virus response. And the way that we chose to tackle this question was using another fish line that was recently published by Professor Eld's group, where they have ISG15, that is a type 1 interferon response gene, tagged with GFP. So you can really see when there is a response, a type 1 interferon response based on ISG15 levels, and we see by the GFP expression. So our first question was, do the beta-2M progenitor cells have a type 1 interferon response? And the answer is yes. So you can see here that the RUNX1, so progenitor cells that express beta-2M, they have higher ISG15. Moreover, when we look at the RNA level, you will also see that the ISG15 positive cells, they have higher beta-2 expression. And when we look at the interaction type, you'd see that the cells that are also green and interact with macrophages, they are groomed. Okay, with that in mind, then we were sure that, well, we're seeing a virus, or at least a type 1 interferon response. What on earth is happening here? So I went back to my compounds because I thought here I have a great tool that I can explore and see what is upregulated once I treat my fish with these compounds. So I took embryos, 3 days post-fertilization embryos, and I treat them with the MSO as control or with my DL-PPMP, that is this ROS-independent compound that triggers CALR on the surface of the cell. And for my surprise, what I found is that in the presence of this compound, there's a huge upregulation of repetitive elements, including endogenous retrovirus. So I thought, well, what if these endogenous retrovirals, that they are also dsRNA by essence, they are somehow going to the cytosol and then signaling through Tol3, and then we have RF3, beta-2M, and grooming. This would be very interesting because, well, endogenous retrovirus and repetitive elements, they are conserved throughout evolution. They would mimic a virus response, so it checks all the boxes. But how can we know that a fish is responding to an endogenous retrovirus? Again, a very lucky time that I had. Well, there was a recent publication where a group by Professor Noémie Hamilton, a group in France, that they managed to create a fish line that the endogenous retrovirus LTR5 was tagged to GFP. And we got this fish and we crossed them with our RUNX1 mCherry and PEG DFP. And we first asked, well, are the cells that have higher endogenous retrovirus also having higher beta-2M as our endpoint? And I hope you can appreciate that, yes, we do see that LTR5 positive cells and here RUNX1 positive cells, they have higher beta-2M. And more importantly, when we overexpress a endogenous retrovirus in our fish, we not only see higher beta-2M, but we also see that the interaction it's more associated with a division. So the stem cells, they divide more. That's a proxy for grooming. Also, when we sort the cells, we see that the the ISG15 positive cells, so the type 1 interferon response cells, they have higher expression of endogenous retrovirus, here depicted by GPC100 and LTR4. So next we thought, can we somehow modulate this? Can we modulate that and try to upregulate? So we have here the overexpression, that's true. But can we do that in a way that we are not affecting development? Because overexpression I did in the embryos, right? So we wanted to do something late in development. And one way that we found to circumvent this question was to use a DNT3 inhibitor. So massive DNA methylation decrease. So the chromatin gets more permissive, and this release more endogenous retrovirus and repetitive elements globally. We show that, and also others. And here we're using a compound named CM272, and we are only treating the fish 3 days post-fertilization. This means that the entire immune system, or most of the blood development, is already in place. So we're not affecting development. We're only affecting, or mainly affecting, this interaction of stem cell in macrophages. And first thing we wanted to see is, well, is this CM272 compound leading to a type 1 interferon response? And this would be like the first thing to see, right? Because if not, then it would be just a side effect of the compound. And in fact, yes, we do see that the fish treated with CM272, they have high levels of ISG15. Here we're looking only at RUNX1 positive cells, so the progenitor cells, they have high ISG15. And when we look at the interaction type, we do see more grooming, as expected. Moreover, when we look at the stem cell proliferation, we also see that they proliferate more, so suggesting that they're having more grooming effect. And where we look at the twister, so the stem cell clonality and how complex it was, then one can say that that's a little bit frustrating, but I wouldn't say so. So when we look at the Gini coefficiency, we don't see differences with the Y type, suggesting that the grooming most likely, or the level of grooming that we're getting here, was not sufficient enough to upregulate the levels of clones in the bone marrow, or in the marrow, sorry, here's a fish, so in the marrow, in the kidney marrow. Nonetheless, when we do the same exercise with the beta-2M crispins, so we deplete beta-2M, then we see that Gini coefficiency goes high, so they're low complexity, and also we start seeing low levels of clones. So this tells us that the endogenous retrovirus, it's actually upstream beta-2M. Beta-2M, it's actually the end point of our cascade. And this for me was very powerful to see. So with that, I come to the summary for my second part, where I hope I convince you that endogenous retrovirus or repetitive elements elicit a type 1 interferon response that promote beta-2M expression. Type 1 interferon alpha responding cells, they have higher beta-2M expression, and they're more groomed, and they proliferate more, and the clones, they propagate. And increase intersposable elements, it's links to type 1 interferon response. And in fact, this comes actually to the end of my presentation. As Diego mentioned, this was recently published. So please, if you have any doubt, go ahead and read. I'll be very happy also to answer questions afterwards. But in a nutshell, I hope I convinced you that homeostatic cytosolic DS or double-stranded RNA prevails dooming of the stem cell. And we believe that is based on a balance between eat me and don't eat me signal, where the eat me signal is provided by CoLR, and the don't eat me signal, it's actually provided by beta-2M. And we were able to find that the molecule that governs both CoLR and beta-2, it's most likely Pol3, and downstream RF3 and beta-2M for the don't eat me. And when there's an unbalance, so when you have high eat me signals, but no beta-2M, then the stem cell is more prone to be doomed, and that clone is eliminated. In the paper, we went one step forward, and we show that the sources, or the major source for this ROS is actually from the mitochondria. So mitochondrial stress is a very highly predictor of a dooming effect. But yes, please go ahead and read. I'll be extremely happy to talk about this later. And in the end part of my talk, I would like to comment on what would be the vision, why we care so much about this grooming and dooming why would be this important in the long run, or why we in the Zone Lab we believe that this can be important in the long run. So basically, if you think about AML, so you have a patient, they have leukemia, myeloid leukemia. You treat them with chemotherapy. The patient gets fine, but there's one clone, a leukemic stem cell, here in LSK, that remains. Over time, this clone can, so normally this clone is quiescent or dormant, but this clone can get reactivated, and then you have an EML relapse. So imagine if we can find molecules that will upregulate CALR, specifically in the LSKs, and downregulate beta-2M. So we will have a powerful way to harness the power of the macrophage that will target to remove the bad clone, because you have high CALR, so the macrophage will come, and you don't have beta-2, so the clone will be eaten. And therefore, you can somehow also decrease the likelihood of AML relapse. And you can say, well, Cecilia, this is sci-fi. I think that's too much to believe that it can happen. So some preliminary data, if I may. No, that's not so unlikely to be happening, because what we observe is that indeed the AML clones, they have higher levels of transposable elements, so repetitive elements. And more importantly, when we sample patients that they have AML, so here we are looking at the same patient. So we have the normal karyotype, the normal genotype, sorry, and the malignant genotype. And when we ask the expression of beta-2M in the same patient, it's the same person, we see that the malignant clones, they express high levels of beta-2M. So highly suggesting that the tumor cells, they are hijacking this molecule to protect them to be eaten by the macrophage. Well, I stop here because I hope you keep tuned in the news in the Zone Lab. I know that there's a postdoc working in this project, and I'm pretty sure that soon he'll be able to share his data with us. With that, I want to thank you all for your attention. I'm very much looking forward for questions and discussions. I also want to thank my mentor, Lenzon. He has been a tremendous mentor, and I'm very grateful for all his inputs, and everybody in the Zone Lab. And if you have any question that pops later, or anything you want to ask me, please contact me by email. I'll be more than happy to be helpful. And that's it.
Great, Cecilia. Great talk, great data. I think everybody here might be impressed by all the work
Thank you so
that you
much.
have done to have this paper published. So we're going to open to questions. We always give preference to the students. So if there is any student with any question, please go on and ask Cecilia.
Also, if I may, if anybody has a question about Roche and how is this transition, I'm also open to answer this kind of questions.
Yeah, that's great. Well, while we don't have questions from the students, I have a curiosity that I would like to ask you about retrovirus, endogenous retrovirus. So you know that there are some publications showing that people with HIV that are under treatment and have no detectable virus, they still have some immunodeficiency. Could there be that their hematopoiesis is impaired because of this antiretroviral treatment, because they're shutting down the endogenous retrovirus as well. That could be impairing the diversity of the clones that come from the bone marrow. What do you think about this?
Absolutely. I think that's an excellent question. It's very interesting. Honest answer is that I don't know, right? There's no data on that. But I would say so. I would say that yes, you can have a clonal diversity being changed. Am I still there? Hello?
Yes,
Okay.
yes.
So yes, I believe so. And also in other type of virus infections, there was some data showing that. So there was a paper recently where they are looking at dengue, so it's an ssRNA, and they're also seeing that CALR goes up, and they're seeing defects in stem cell clonality. So no, I don't think it's something only about endogenous retrovirus or LTL4. I think it's something really very ubiquitous and global. So yeah, absolutely. It's a great question.
Great. Well, and great work, right? So Gustavo and Miriam both have questions. I think Gustavo was the first one.
Okay, Cecilia, super thanks. It's very wonderful, intriguing data and concepts. And I am a bit confused with a couple of things.
Sorry.
First, do you believe that all progenitor cells are engulfed by macrophages? So I just want to, it's brand new, and I'm thinking what would be what would be the function or the evolutionary pressure to this, okay? And when you say eat me and don't eat me signals, it seems to me that in both cases, the cells are being engulfed anyway. In one is like grooming, so the cells result in live cells, and the other is dead cells. So please, if you can explain me a little bit more, what are the differences?
Absolutely.
And whether I know that I'm a bit confusing as well. So I think let's come back to the first. All cells need to be engulfed.
That's a great question. No, so the full engulfment, so the full eat me process, you doom the cell, so you have no clones.
Right.
So if you overexpress ROS, for instance, you also affect the stem cell clonality, but on the contrary, because you eliminate them all. So in a parallel part of my project, we have fish that had lower expression of FOXO3A. So they have high levels of ROS, so stress cells, and the marrow was also very, very low in complexity of clones, but not because they were not expressing the protection molecule, the beta-2M, but then on the contrary, they were so stressed that this was a signal to take them out. So no, it's not every cell that is engulfed. Otherwise, you would have nothing in your marrow at the end of the day. And also, not all the cells are in interaction with macrophage. Nonetheless, we see 70% of the cells, of the stem cells, being in close contact and interacting with the macrophages. And what we see is that the cells that are groomed, so with this partial nibbling of the cytosol, they proliferate. So there is a division, and the clones propagate. So you have a continuation of that clone. We also have long interactions that leads to nothing. So they just stay there, but there's no grooming, there's no dooming. Macrophage goes out, but then the stem cell does nothing. It stays there. It doesn't really proliferate. It's just a stem cell. It stays as it is. So we believe that this interaction, it's important for stem cell differentiation, proliferation, for the grooming point of view. And for the dooming point of view, we believe that this process, it's important to really get rid of the stress cells or the clones that they can be harmful in the future.
Okay.
Also, what we found, and this is more into...
No, go ahead.
Okay, it's just that you ask about evolutionary point of view, right? So this is a little bit in the exploratory speculation, but what we thought is that maybe this mimicking a virus infection would be relevant for innate immunity, in the sense that if you have a constant or a tonic increase of this type 1 interferon, you could change the lineage and you'd be more protected against something. And what we found is that when you treat the fish with poly-I poly-C, we see an increase in neutrophils. So this kind of suggests that maybe it's a tonic expression that boosts your innate system and you would respond more against pathogens. But that's in a speculation, it's more. There are some papers that tackles and talk about innate immunity and train immunity and how type 1 interferon it's important in humans. So that's why I said it's a little bit speculation and bringing here and there, but it can be that there is a link between neutrophil differentiation and this tonic expression of endogenous retrovirus.
Right. So, but in the whole scenario, it looks like the macrophages then controls the process, either in a tonic way or in an activated way. So that's what I'm thinking here. So it seems to me a bit like the thymocyte selection, where you have the cells, a compartment of cells that are ignored. In the T cells case, they die. In this case, they just don't do anything. They're waiting. And then you have a negative and positive selection. Okay, so if the cells are stressed, they are negatively, then they die. And then the other one. So it's a way to control the whole system. And you mentioned poly-IC, but what about things that like cytokines that induces hematopoiesis? Does these cytokines change as well this?
So what we were able to see, and this was not my project, but actually Sam's. So he found that when there is this grooming interaction, this grooming type of interaction, the macrophage express a type I1 beta. And I1 beta is important for stem cell proliferation. And when he blocks this I1 beta, then he doesn't see the clonality going up or changes in clonality. So the cytokine that we are sure that plays a role here is I1 beta.
What about GM-CSF or one of those that
the classic ones, right?
Drive the proliferation, you know?
Yeah, this would be excellent to test it. We haven't. Most likely, I would say it would promote stem cell division and stem cell differentiation, because you're basically kicking them from the cliff, right? And then you're doing this in a non-endogenous manner, and then you don't care about macrophage dooming grooming. You just go.
So sorry, just one quick thing. As far as I'm aware, you know, maybe I'm completely wrong. Beta 2 microglobulin, this molecule is expressed alone? Because to me, it's expressed in the context of MHC class 1.
Class 1. Yeah, so it's mainly expressed in the context of class 1.
Okay, so does any of these inhibitory receptors for class 1 plays a role?
Yes, LRP1, LIR3, LIR6, and 3, we believe that plays a role.
Okay, so maybe beta 2 is just a marker of MHC class 1. If you knock out MHC class 1, do you have the same phenotype as beta 2?
Yeah, correct. Yeah.
Okay, so then beta 2 is just the carrier. MHC is the important one, or not?
Not necessarily, because not necessarily. We believe that beta 2M is the don't eat me molecule, because it's where you have the docking. So you have class 1 as a full. And again, this comes with a lot of speculation and things that I would love to tackle in the future, where we should also look at new antigens being expressed and then being presented, and a whole new world appears in front of us. So we believe that class 1 and beta 2M, it's important for this don't eat me signal, but beta 2M is the molecule that drives the don't eat me, because it's where the docking happens. If you knock out class 1, yes, you still have beta 2M in the surface, but in a very low expression level. It's really not really there, and the turnover is also quick. So that's why I said not really.
Right, but it's just that the evolutionary controlling cells that are MHC class 1 negative is very important. Okay.
Absolutely. Yeah.
So that's what I meant in the evolutionary aspect. So maybe this is meant to check whether the cells that are populating the blood are MHC class 1 positive. Okay, this is complete. It's very important.
Absolutely. And just to tack on this, in the mouse, when we treated them with poly-I-poly-C, we look at class 1 molecule, actually. We didn't look to beta 2M, because the antibody is way better. And we see that it is increased. So yes.
Thank you.
You're very welcome. I hope you're less confused.
So Miriam also has a question for Rio.
Yeah, thank you, Diego. Thank you, Cecilia. We have a couple of questions here. I'm going to ask one and then I'll pass it on to Bruno. One of them continues on what Gustavo just said at the end. I wonder if in the fish you find a transmembrane portion in the beta-2M, because as long as I know, in mammals you don't have, so it wouldn't just lay on the membrane. But I wonder if in the fish there's some residual portion of the molecule that could be retained such that it would stand alone in the membrane even for a short amount of time, because as far as I know, it would just float away if it was sent to, on that matter, right? So that was on the, I guess I was a little bit confused as Gustavo, because of that, as long as I remembered, if it went to the membrane, it would just float away as a cytokine because it wouldn't have anything to anchor it to the membrane.
Yeah, it's exactly what you said. It is there, but the short time, it's very, very low. It stays for hours or so. So the staining, we never managed to have a proper, decent, long life staining. Ergo, I'm always choosing class 1, especially in the mammals. So there are, in the literature, some papers that shows that, yes, it can stay in the membrane for hours, but then it flows away. So the turnover is very, very fast, but it is there. If it's enough time, I don't know.
Okay. And one thing I think I just missed, the endogenous RNA that is expressed, is it in the stem cell, the macrophage, or both cells?
Excellent question. So what we found is primarily in the stem cell. So when we do the RNA-seq, the bulk RNA-seq, we sorted for RUNX1 positive cells, so not stem cells, but progenitor cells. And also when we look at ISG15, it was mainly focused on the RUNX1 positive cells, so stem cells. That's true. And then again, here's extremely speculation, things I would love to do in the future. What I found is that when there's this grooming effect, so grooming phenotype, and we have the ISG15 signaling, you would see that when the macrophage is grooming, there are some particles, green particles, that enters the macrophage as well. And these I don't know what's happening. Why this signaling, this type 1 signaling molecule is entering the macrophages, and what's doing there? Because maybe it's also changing something in the macrophage. Maybe this IL-1 beta production comes from this signaling that the macrophage just took it from the stem cell. But this is pure speculation. We have not tackled the macrophage in this project at all. It was mainly stem cell, and all the endogenous retrovirus that we found and type 1 interferon response was in the RUNX1 positive compartment.
Okay, thank you. So, Bruno.
No worries.
Right. Thank you very much for your talk. It was very exciting. I promise I will read all the relevant papers so I can learn a little bit more. It was very, very nice. But my understanding is that or what you showed here was mainly focused on the clonality of the stem cell. But do you have data on what type of cells were actually being produced? Does it change the output of the bone marrow in terms of type of cells? And also, if these grooming interaction actually is different according to the fate of the stem cell, or the differentiation path that the daughter cells will eventually follow after signaling, after this interaction.
Great questions. So for the first one, we only have a glimpse, a very small glimpse on what's happening there, which is when we treat the fish embryos with poly-IC, we saw an increase in neutrophils production, so suggesting that this type 1 interferon could somehow be associated with a granulocyte differentiation, or boost this differentiation. About the second question, if grooming would lead to like more macrophage, more myeloid cells or lymphoid cells, it's very hard to tackle this. What I can tell you is that when we deplete or when we decrease grooming, right? Because again, I can talk the other way around, right? So when we decrease grooming, we don't see changes in the myeloid and lymphoid compartment in the sense that everybody goes down, but we don't see that one takes over. So we believe that is not affecting one lineage specific. It is mainly affecting the stem cell fate in the sense that it proliferates or not. It continues in there and propagates, or it's doom and the clone is not there anymore. I hope this answers your question, but we cannot really follow the specific cell and see what it becomes. We see that in the end, there's no global or significant difference in the lineages.
And do you think that these grooming behavior, if modulated, it can be used to modify how effectively bone marrow transplants are actually they actually work?
Yes, so it's funny that Adeline always tell us that in a project you have to have two questions. One that you can answer and the other one that you can dream of. So I think we managed to answer that beta 2 and color are important for dooming grooming. What we can dream of is that we can use dooming in our favor to eliminate these bad stem cell clones and have less AML being relapsed. And we can use the opposite, which is exactly what you said. So we can use its DL-PPMP that increase beta-2M before the transplants and then maybe increase the engraftment. I didn't test that myself, but yes, these are the two dreaming questions that we would like to answer. If we can use dooming to AML or myeloid disorders and the grooming to enhance xenografts and transplants. Yeah.
Right. Thank you very much. I look forward to what will be coming next.
I'll be very happy if you can test the DL-PPMP and tell me if the transplants they're better. I'll be extremely excited to hear that.
Sure. I don't think we have other questions here on our side. So Diego, Gustavo.
Okay. So I don't see any questions popping up on the chat too. Right? So as Cecilia told you guys, you can contact her directly if you have further questions, right? Thank you again, Cecilia. It was amazing. Very, very nice data.
Excellent.
Very good.
Thank you for the opportunity. I'm very much honored.
Yeah.
All right.
Okay.
Thank you very much, Cecilia.
Yeah, super thanks and
Bye-bye.
wonderful week to all of you guys.
Bye, guys. See you.
See you. Bye. Thank you.
Have a nice one. Bye.